So, we know that enzymes are catalytic proteins, and we know that they are capable of increasing the rate of metabolic reactions in living cells. Let's take a minute and think about what enzymes are doing with regard to that reaction rate. Now, on this slide, there's a very busy looking graph. Don't let that distress you. We will talk through this graph together. On the x-axis, you can see basically time going by. This represents the progress of the reaction over time. On the y-axis, we see what's called free energy. I want you to think about free energy as being the energy that is available to a cell to do work, to get things done. This graph represents a reaction that involves a molecule called A and a molecule called BC. And you can see there's a covalent bond holding B and C together. Those are our substrates or our reactants. And those molecules, like all molecules, have some energy associated with them, some energy sort of stored up inside of them. You'll notice that there's this red line here, hits that y-axis right there. That's the amount of energy that's inside these substrates, A and BC. And if we follow the red line across time, you can see that something's happening here. If you follow the dotted line, something's happening here. The free energy is going up, 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 until it hits a very high level, if you trace it over to the y-axis, that's labeled the transition state. Now, look at the substrates at the transition state. They look different. Old bonds have been broken down, but no new bonds have been formed yet. That is what we call the transition state of a chemical reaction. It's a very high energy state. Now let's continue to follow the red line. You'll notice it drops down precipitously until we see products made. And our products are now AB. You can see a new covalent bond right there, and the newly freed up molecule called C. So if we were to take this line and trace it back over to the y-axis, we are at a lower level of free energy than we started. In other words, from the start with our substrates to the end with our products, some energy has been released. And of course, that tells us something. That tells us that this is an exergonic reaction. This is a catabolic reaction. Now, I want you to look at the difference between the dotted line here and the solid line here. The difference between these two lines is the addition of an enzyme. If you start with the free energy of the reactants, if you were to draw that line all the way across, the distance, the difference between that line and the top of the peak where the transition state occurs, that difference from here to here is referred to as activation energy. This is the energy that you have to take. This is the energy that it requires to reach this critical transition state. Now, if you put an enzyme into the mix, what's going to happen is your activation energy, which is signified by a capital E and a lowercase a, your activation energy is going to drop quite a bit. See with the solid red line that I can hit the transition state at a much lower energy level. If you trace it over to the y-axis, I'm at a much lower level of energy, and yet I'm at the transition state. Another way to think of this is that when an enzyme lowers the activation energy of a reaction, what it's doing is it's making it easier to reach the transition state. You can hit that transition state with less energy, so it will occur more readily. And that means we will be able to drop down and make product more readily as well. Enzymes lower the activation energy of a reaction. Now I want to mention one other thing, and that's this little symbol of delta G. I don't want you to worry about delta G. That's something we're not going to be talking about in this course. I just want you to know what this EA stands for. That refers to activation energy, and it's the energy required to hit that critical transition state. And when we put an enzyme into a reaction, we're going to lower the activation energy. It's going to become easier to hit the transition state, which means we're going to make product more rapidly. So, to summarize, the activation energy is the energy needed in a chemical reaction to reach the transition state, essentially halfway between substrate and product, where the old bonds have been broken down, but no new bonds have been made yet. Without an enzyme, the activation energy is typically quite high and hard to reach. But when you put an enzyme in there, the activation energy will be lowered, so it will be easier to reach it. Product formation will occur more rapidly, and the rate of the reaction will be increased. In other words, the product produced per unit of time will go faster. Enzymes are critical to metabolism. Enzymes are what allow the vast majority of biological reactions to occur in real time. Cells are busy little creatures. They need metabolic reactions to go on in their own time, in their real world time. Enzymes are powerful things, therefore. And a cell would not want to lose control of an enzyme. So, just as most biological reactions require enzymes, enzymes are tightly regulated. They have to have control of these powerful molecules. And the two most common forms of regulation are what's called competitive inhibition and allosteric inhibition. Let's talk about these regulatory mechanisms. Let's begin with competitive inhibition. You've heard me say before that cells are thrifty, that cells don't do things unless they have to do them. Cells don't waste, in other words. Now, if a cell has an enzyme that it needs at certain times, it's not going to allow that enzyme to function when it's not needed, because that would be wasteful. It's not going to allow that enzyme to make product that is not needed at that moment. And so cells control enzymes sometimes by producing what's called a competitor molecule. A competitor molecule is going to be very similar in size, very similar in shape to the substrate for that enzyme, because that competitor has to be able to bind to the active site. Now, here's where it's controlling for the enzyme. The competitor molecule, it's going to be able to fit in the active site, but it's not going to be able to take part in that chemical reaction that the substrate normally takes part in. In other words, it's going to bind to the active site, and it's just going to block it. It's going to prevent the actual substrate from binding, and so it's going to prevent the chemical reaction from occurring. Take a look at the drawing at the bottom of the slide. This green blob down here represents an enzyme, and you can see there are these concave regions here and here and here that represent active sites. Here's our substrate molecules. There's this orange one here, and then there's this red one here with a covalent bond in the middle. Remember, the substrates will fit perfectly in those active sites. Now, the cell wants to basically shut off this enzyme temporarily, and so it creates a competitor molecule, and that's what you're looking at in blue. This regulatory molecule is very, very similar in size and shape to the substrate, and so it's able to sit in that active site or those active sites. Now, it's not the substrate, so there's not going to be any chemical reaction going on when it comes in and binds to the active site. But because it's sitting there, it's going to block the actual substrate from coming in, and it's going to therefore block the chemical reaction from occurring. Now let's talk about allosteric inhibition. Allosteric inhibition also involves one of these regulatory molecules. The difference between competitive and allosteric inhibition of an enzyme is simply where the regulatory molecule binds. The allosteric inhibitor molecule is going to bind somewhere other than the active site. And you might say to yourself, well, how's that going to work? How is that going to prevent the substrate from coming in and binding to the active site? And the answer is, when the regulatory molecule, shown in blue here, when it comes in and binds somewhere else on the enzyme, it's going to cause to happen what always happens when you bring two molecules together. It's going to cause that protein to change shape. And when it changes shape, it will alter the shape of the active site, even though that's somewhere at another location on the enzyme. Remember, any time that you stick a molecule onto a protein, it doesn't matter if it's a big molecule or a little molecule. Anytime you stick a molecule onto a protein, you're going to cause that protein to change shape. And in this case, it will be a significant shape change in the sense that the active site is going to be affected and the substrate simply won't fit. The active site is open, but it no longer fits the substrate perfectly. So this diagram right here on the right, this represents allosteric inhibition of an enzyme. I do want to point out that it is possible for a cell to use this type of allosteric mechanism to activate an enzyme as well. It is possible for a cell to create a different type of regulatory molecule, shown here in yellow, that when it comes in and binds to the enzyme and causes the enzyme to change shape, it will go back to the shape that allows the substrate to bind to the active sites. So there is what we can think of as allosteric inhibition going on, and there's also some allosteric activation going on inside living cells. One other thing I want to point out before we leave this idea of enzyme regulation relates to competitive inhibition. A lot of the drugs that we use in medicine are competitive inhibitors. The drug known as a non-steroidal anti-inflammatory drug, or an NSAID, something like ibuprofen, that's a competitive inhibitor. and it is a temporary competitive inhibitor, as you would imagine. In other words, that inhibitor can only come in and bind to the active site like this for a short period of time. After that, the competitor will either be broken down or it will be removed from the active site, and the inhibition will end. There are molecules in nature that are competitive inhibitors, and they are permanent. A good example of this is the molecule called cyanide. Cyanide is a competitive inhibitor for a critical enzyme in the body, but when cyanide binds to the enzyme's active site, it binds permanently, and the person dies as a result. So it's important to know that there are temporary competitive inhibitors, and those make good drugs for us in medicine. But there are also permanent competitive inhibitors that we would think of as toxic. In addition to a cell being able to regulate its enzymes, it's also important to know that there are environmental factors, there are conditions inside the cell that will influence the activity of an enzyme. The two critical environmental factors I want us to be familiar with are temperature and pH. Now we need to get into a little bit of chemistry here as we talk about these two factors. Temperature is a measure of the kinetic energy of molecules. So if you recall from your chemistry class, all molecules are actually in motion on our planet. So it doesn't matter if you're looking at something like your cell phone that seems to be completely inert and immobile. The molecules inside the materials in your cell phone actually have motion. Now, once you drop down to a temperature called absolute zero, if that were possible, all of the molecules would stop moving. But above absolute zero, there is some degree of motion, and that's what we refer to as kinetic energy. So, the higher the temperature, the more motion, the more kinetic energy. The lower the temperature, the less motion, the lower the kinetic energy. So, if the temperature conditions change in a cell, that's going to change the kinetic energy of the molecules in that cell. That's going to change the motion of both the substrate and the enzyme. And you might say, well, why does that matter? Well, it turns out that motion is important for bringing the enzyme and the substrate together. We need them to collide with each other in order to get that substrate to bind to that active site. The higher the temperature, the more collisions are going to occur. The lower the temperature, the fewer collisions are going to occur. It turns out that all enzymes have an optimal temperature for their very best performance. Now let's think about pH. Recall from chemistry that pH is a measure of the concentration of hydrogen ions that are in a solution. Now, as you increase pH, you make a solution more basic and you reduce the concentration of hydrogen ion. As you lower pH, you make a solution more acidic and you increase the concentration of hydrogen ions. Hydrogen ions will interact with protein. Hydrogen ions will come in and bind to enzymes. So by altering the concentration of hydrogen ion in a solution, you're going to alter how much binding of those hydrogen ions is going on with enzymes. And depending on how much hydrogen ion is sticking to those enzymes, that may or may not cause the enzyme to change shape in a way that either makes it more active or less active. Remember, anytime you stick something onto a protein, you're going to cause that protein to change shape. All enzymes have an optimal pH for their best performance.